Hi everyone, I'm Mohammed Cherlu. I'm here to explain how you can use our SSVEP toolbox in your own BCI projects. Recently, we published a review paper that covers 19 template-based frequency detection methods in SSVEP-based BCIs. These 19 methods are categorized into 4 groups: CCA, MSI, CORRCA, and TRCA. In this paper, we provided a clear description for each method, what is their goal or how they detect SSVEP frequency, and also we provided a clear flowchart that helps researchers to implement each method. Furthermore, we provided a MATLAB code for each method, so you can easily use this code or customize this code for your own dataset. In this paper, we wanted to evaluate all methods under the same condition to see which method is good or which is not good. To be brief, we wanted to understand their pros and cons, and we conducted several experiments, and I highly recommend you to study this paper, especially the experiment 1, because we revealed 4 factors that are crucial to create an optimized SSVEP frequency recognition BCI. For evaluation of the methods, we used SSVEP benchmark dataset, which is a 40 class dataset and was recorded from 35 subjects. So these codes for each method are implemented based on this dataset. So if you want to use these codes, I highly recommend you to first use the codes for the SSVEP benchmark dataset. Then you can easily, after you get familiar with the code and with the idea, with the, everything that is related to the approach, you can easily customize it for your own dataset. However, here I'm gonna provide some further explanation to help you understand the codes and also easily customize the codes for your own dataset. Okay, the first thing you need to do is to come here to my GitHub channel and download the SSVEP toolbox. As you see, we have many files here. Some of are named like demo file. These are the implementation of each approach. As I said, we implemented 19 SSVEP frequency detection methods. So their implementation are written here, are coded here, so you can use these demo files. And also we have a file named dataset. However, it is empty. You need to download the dataset or put your own dataset here and read each one and apply the method on that dataset. And also we have SSVEP toolbox functions. We wrote a function for each base model and some other methods that are used in these demo files. As you can see here, we have CCA, CORRCA, MSI, MCCA, and everything in here. You can see that there are some functions that are used in these 19 approaches. We have another function named my sine cosine reference .m. This one is used for creating pre-constructed sine cosine reference signals in SSVEP. I consider that you are familiar with the content and you know that what's going on in SSVEP frequency BCIs, frequency detection BCIs. So I just briefly explain that how you can use these files. Okay, the first thing you need to do is that to download these files, and if you come here, you can see that we have a readme file, and if you read this file, you can easily use file. However, I provided this video to ensure that you can easily use the file and also customize it. Okay, first you need to download the SSVEP toolbox, unzip it in your direction, in your laptop or in your PC. Then you need a dataset to apply this method on that dataset to see its performance or whatever you want to do. Okay, the next step is that you need to download the benchmark dataset. As I said to you, to evaluate the methods under the same condition, we use the benchmark dataset. This is a 40 class dataset and it was recorded from 35 subjects. So you need to go to this website and download it. Here you can see that there are 35 subjects and the file are here. You need to download all the information related to this data set. Then you need to download this tensor toolbox. Some methods like MCCA, MCCCA, and multi-base CCA use tensor-based calculation. So if you want to use these methods, you need to go here and download this toolbox. The link, if you click on the link, you will go here and you can download these codes and put here. As you can see, let me come here and see. You need to put it here after you download the file. You will see in my PC that I downloaded and I have inside this file. So first you need to download this toolbox, then download the data set, and also if you want to use these methods, you need to download this GitHub file too. Then after you download everything, you need to add this toolbox to your MATLAB path. As I didn't say, but I need to say that these codes are implemented in MATLAB, so you need to add this toolbox to your MATLAB path to be able to use them whenever and wherever you want to use these codes. Then you can easily run each demo code to see each SSVEP frequency detection on each detection performance on each subject. So I downloaded all of them and I have them here. Let me go here. If you can see, here's the SSVEP toolbox. After you download it and unzip it, you will see these files. These are the demo files for each method, for each approach. And this is the toolbox that have that tensor toolbox. As I told you, if you want to use MZ, MCCA, and something like that, you need to add this file too. And also we have functions here. As I told you, we have implemented these base models like CCA, CORRCA, TRCA, MSI, and other functions that are used in approaches, and also this function. Okay, first we need to install these files or add these files into our MATLAB path. So after that, in every direction or every time we want, we can use these files in our codes. So the next step here is that you need to download the dataset. As you see, I have the datasets and I put them here, 35 subjects. If you go inside them, you see that there's a MATLAB file for each subject. Okay, so you need to download the dataset too, and now we are ready to use the files. Okay, if you want to add them temporarily, you can add manually these files into your MATLAB path, but if you want to add them permanently, so you can run this code, it can take care of everything and add the toolbox in your MATLAB path. Okay, let's go here and see what's gonna happen. Okay, first I'm gonna open install file, run it. After running this code, all the functions or files in SSVEP toolbox will be added to our MATLAB path permanently. Okay, I ran the file and now they are added to our MATLAB path. So now we are ready to use or run these files. First, I highly recommend you to run all of these files on this data set, the benchmark data set. Then you can customize each demo code for your own data set. First, you need to get familiar with the code, with their structure, and also I recommend you to read the paper, because these codes are exactly coded based on our description or our flowcharts in the paper. So first, read the paper, then come here and run codes on this data set. After that, you can customize it for your own data set. Let me go here and see, run one of these demo codes. The simple one is standard CCA. Okay, I assume that you are familiar with this code or other codes and you read our paper. I just wanna explain some parameters to make sure that you can change them whenever you want to change them. Okay, the first parameter is FAS, or sampling rate. For this data set, it is 2050. Sorry. This data set is, the number of channels in this data set is 64, and in most papers people use just 9 channels. Their name is written here, and I wrote the number of those channels here. So if you want to use these 9 most informative channels, you can uncomment this one and comment this one, or if you want to use all channels, you comment this line and uncomment this one. Therefore, all of channels will be used in SSVEP recognition process. Okay, I'm gonna uncomment this one and use these 9 useful channels. Okay, this is the first one. The second one that you need to change, if you want to use other channels, you can go and read the file, channel location file, and get familiar with the number of channels and name of channels, and then you can change the channels if you want to change them. And the data length in this data set is 6 seconds. However, the first half and the last half is not related to SSVEP. So when you want to evaluate your method performance on a specific data length, for example, you want to evaluate your method's performance on 1 second data length, therefore you need to crop or segment the dataset from first half until 1.5. As I said, the first half and the last half is not related to SSVEP. Only the middle 5 seconds is related to SSVEP. However, we do not use all 5 seconds. In some studies, we use just 0.5 seconds or 0.1 seconds or 1 second, 2 seconds, 1.5 seconds, 2.5 seconds, and every number can be here. It is related to you. So if you want to evaluate your method, you need to evaluate its performance on different data lengths. So here you can easily change it. The duration is based on seconds. If you define it as 1, in here our code will segment the dataset from 0.5 till 1.5. If you define it 2, it will crop the dataset from 0.5 till 2.5. As I said to you, the first half and the last half is not related to SSVEP, so in this dataset we need to crop the dataset from 0.5 and until the last half. Okay, here it can take care of everything. You just need to define your data length. For example, if you want to define it 0.5 or 0.1, you just define it. Okay, I wanna put it 1. And another thing that we applied on the data set is a bandpass frequency filter. For this data set, 5 harmonics are informative. Therefore, we filter data set with the frequency range from 8 to 19. Again, you can change the frequency band. Okay, another thing here is that each stimulus has a frequency and their information are inside this file. So if you use this data set, do not change anything. But if you have another data set, you need to define the frequency information of each stimulus and put it in the file like this. Okay, I created labels for all of these trials. Let me give you another information here. Okay, this data set was recorded in 6 runs, and in each run 40 trials were recorded, and for each stimulus there was 1 trial, and these orders are the same in all 6 runs. That's why I define labels from 1 to 40 and repeated 6 times. Okay, from now on everything is okay. If you have another data set, you need to define based on your data set information. Okay, another parameter you need to change is the number of harmonics in creating sine cosine reference signals. Based on the equation, we need to create some sine cosine reference signals. If your data set has information until harmonic 5, you need to consider this one 5. Or if you think that your data set only has in two harmonics information only has information in two harmonics, just consider 2. This is optional. You can change it based on your own data set. In this data set, it is proved that the information the data is informative until 5 harmonics. That's why we considered 5, and also here based on that information we create such filter that covers all harmonics in the data set. And here you just define the number of subjects. I created a loop, and in each iteration one of subjects is loaded and the frequency detection based on the standard CCA will be run, and the frequency SSVEP frequency of each trial will be recognized based on this algorithm. You don't need to change anything here until you want to customize or develop these algorithms. Okay, the information you need to change. The first one is sampling rate frequency, channels that you want to use in your study, and also the duration of the trial. As I said, in order to evaluate your method, you need to evaluate its performance on several data lengths. So you can define it 0.1, 0.2, 0.3, and 0.5 until 5 seconds in this data set. The maximum data length here is 5, and so you can change it and evaluate the method's performance on that data length. You can also change the frequency range in bandpass filter and the number of harmonics in creating sine cosine reference signal. So after that, this code will take care of everything. It will load the data of each subject and apply the approach on that data set to recognize the stimulus frequency of each signal. Okay, let me run it and make sure that everything is okay and it can run on the data set. As we can see here, the code is running and it's okay. Okay, I wanna interrupt it and go to another code just to show you that everything is okay. Let me run my favorite algorithm, filter bank ensemble TRCA. This is my favorite one, and also data driven CCA is another one that I really like. They are so powerful methods. Okay, the duration is 1, and okay, everything's okay. I'm just gonna run this file. If you want to know what are they or how they work or what is their approach, go and read our paper. I highly recommend you to read our the experiment part, because we revealed that 4 factors are crucial in designing an optimized method in SSVEP frequency detection. As you can see, for the first subject, we have almost 100% accuracy by filter bank ensemble TRCA, or the other one is like that. For CCA, I think we got 35. There's a meaningful difference or significant difference. This method works phenomenally, works very well, and I recommend you to study this method thoroughly, and also the data-driven CCA. They are somehow the same algorithm, but they are in their theory or in the implementation, they use a different approach to detect SSVEP frequency. However, in reality, they act the same, and we showed it, or we proved it in our paper too. Okay, that's it. I explained that how we can use our code, and again, I'm gonna recommend you that first read the paper, then run these codes on the benchmark dataset, get familiar with the dataset. If you want to customize the code for your own dataset, first understand the benchmark dataset and see that how these codes are written based on that dataset to process the information of that dataset. After that, when you get familiar with the dataset and the demo codes, you can easily customize the codes for your own projects. Okay, I think I explained everything that was needed. Now I'm gonna go and see you next time. Bye for now.